So I want to start this last Ask the Experts question on time because some of our panelists, we're going to have to catch flights, so we'll sort of move this along expeditiously. First, those of you who were not in the cardiac session in the breakout room, probably have not heard Dr. Beth McNally before. She's both a researcher and clinician at Northwestern University here in Chicago. And she is one of the people involved in a new and in an additional clinical trial that just was posted a couple weeks ago on clinicaltrials.gov. And Beth, can you briefly describe that? That's for both Becker MD and LGMDs.
Yeah. We are doing a weekly steroid trial, and it's supported by the Kurt and Peter Foundation, so thanks, Kurt and Peter Foundation. And the goal is to just test a dose of once weekly steroids, and we have a number of LGMD subtypes that we're including this. And I don't know if Aaron's still in the room, but he's our recruiter, and he's here. I think he's waving over there. And so it's basically involves just taking steroids once a week. We're mainly doing this. It's all open label, and it is designed to just look at safety at this point to make sure that it's safe to do this. And we will do an MRI at the beginning, at the end, and the end of 6 months, as well as biomarkers at the beginning and the end of 6 months to look at skeletal muscle function with the MRI and to look at just signatures of safety with the biomarkers. So that's recruiting. I think we're getting there. We're maybe a third of the way in recruiting. So 15 LGMD and 15 Becker patients, and it's being done here in Chicago.
Yeah, and it's now posted on clinicaltrials.gov. And so the advantage of weekly steroids, and the Jane Foundation has actually done a study in dysferlin deficient mice. If you give mice steroids every day, they have a lot of the same side effects as humans, but if you give it to them once a week, their side effects seem to be pretty much non-existent. So that's sort of the rationale that Beth has discovered in her research in animals and now being translated to people. Okay, so we're going to start with some questions about gene therapy, and so there's a couple on the redosing issue, and I know Jeff alluded to this in the talk, but so there's one question on, well, what if I'm in a clinical trial at a certain dose, and then they figure out that receive gene therapy, and then they discover that the optimal dose is, something higher than I actually got. Is there any way I can, get the appropriate dose? Or what happens, if I get it's approved gene therapy, and then it stops working after some period of time? So, big questions. Anyone want to try tackling those?
Well, I can say a few words about it. I'm sure my colleagues will have things to add to that. It's a big issue, and there's a lot of unknowns. I mean, the big unknown is even for a patient that gets an optimal dose of gene therapy, we don't know how long it's going to last for. It's probably going to last at least a decade, hopefully longer. But it may vary with the disease and with the gene therapy regimen and things like that. Perhaps equally, if not more important, are the patients in the early phase clinical trials that get a low dose. What about the prospect of coming back and getting a more optimal dose? What's going to happen with that? About all I can say is that it's an important issue and something that is well recognized in the field and is being heavily studied by a lot of different laboratories around the world. That's the optimistic point of view. The pessimistic point of view is, at the moment, we don't really know how to do a second dose. The problem is that when you receive a gene therapy treatment, you are being vaccinated against the gene therapy drug, just like a vaccine against measles and things like that, and that prevents you from getting a second dose. However, there are, for every problem, there is a solution. That's what really science is all about, is identifying problems and figuring out how to solve those. One of the things I alluded to in my talk, when I first started talking about doing gene therapy 30 years ago, people said two things to me. They said, muscular dystrophy is not a curable disease and gene therapy will never work. Well, we know that muscular dystrophy is curable. We're seeing it starting to happen. We know that gene therapy is starting to work. Those are problems that could be solved. How do you go about overcoming this vaccination effect? It requires looking at the body's immune system, trying to suppress it, trying to remove the components of the immune system that prevent readministration. There's labs. There's a big lab in Paris at Genethon. There's labs at the University of Florida that are doing a tremendous amount of work in trying to figure out how to suppress the immune system and do a second dose. There's some groups of researchers in Seattle at the University of Washington, where I'm at, that are doing the same thing. So far, they actually have encouraging data, at least in mice and in some larger animal models, that you can, under the appropriate circumstances, give a second dose. But these treatment regimens are not quite ready for prime time. They're still being developed, but I'm optimistic that in the next few years it'll become possible to do a second dose.
Can you hear me? Yeah. Just to add a few things to that, I think Jeff summarized it very well. The reason that redosing is a problem is because the antibodies that your body raises to the virus, and there are several ways to eliminate those. So we've been working on a way using plasmapheresis to remove these antibodies from the bloodstream. So we did a study about 5 years ago now in large animals that had positive antibodies already, that they were already immune to the virus. We removed those with plasmapheresis and were successful. And then we're trying to apply the same technology. Others are trying to use strong immunosuppression to either suppress the antibodies after or suppress them in the first place. So there's collectively a lot of groups trying to work on this. It's been known as a potential problem that we needed to solve very early on, and so there's a lot of work being done to do that. So it's definitely a high priority for all of us.
Okay, so there's a question specifically directed to you, Louise, and that is, we've seen the biomarker or protein expression data for the 2E trial. When are we liable to see? Well, first, what's the correlation between the biomarker and the functional data? How well established is that? And when are we liable to see functional data?
Sure. So we're close to, why it keeps going on. We're about 9 months out for the first group of individuals. We'll be having, we're analyzing the functional data from that group soon, and we'll release those results soon. The endpoints that we've identified and that are listed on clinicaltrials.gov are things like the 100-meter, the 10-meter, the North Star assessment, similar functional tests that we use in Duchenne. And so those will be critical, and then we'll be dosing our higher dose cohort, which will be informative in selecting the dose for the pivotal trial. So soon.
Soon, okay.
Soon's the answer.
Okay, I think we're all looking forward to that, is safe to say. Okay, so I think we've already seen one example. What about patients who don't live in the US or a country where a clinical trial is happening? Can they still participate? I know Alexandra and his family came here from the Netherlands, but can anyone discuss international recruitment a little bit?
Sure, I can. For the domaguzumab study, we had 2 patients come from Germany, and their travel funds were covered by the trial. I'm doing a trial with catabasis in Duchenne. We have several coming from the UK. Their travel is funded by the trial. So I think that's becoming more common, that these trials are open to international participants, and the sponsors is covering the trial costs.
Yeah, I guess, I'll add a little bit of my own thought. In a rare disease, recruiting for clinical trials is very challenging because it's a rare disease. And I think there probably is more willingness to, find patients wherever they might be within reason. Is that? So kind of a general thing about clinical trials, do they help us now, or are they mostly for the benefit of future patients? It's kind of one of these big philosophical issues. Anyone want to jump in?
Well, just a general comment about that. I can say that every clinical trial we've ever done has taught us something that was applied to the next clinical trial. That goes first for recognizing the problems with AAV antibodies and discussing with the FDA the exact amount of neutralizing antibody that was acceptable. For example, in one of the first trials we did in LGMD 2D, we found one patient had a titer of 1 to 3,200. The FDA then ratcheted us down to 1 to 50, and that has been applied since then, except for our new microdys trial where they allowed us 1 to 400. So lesson learned was about AAV antibody and how much was tolerated. We also learned from a DMD trial that if we express the gene into an area of deletion, we'd likely get an antibody for a new protein that was produced and not recognized by the patient. So we learn things every step of the way, and it's a good lesson. It's unfortunate that we have to learn. We wish we knew everything going in, but it's unequivocal, and that's why we thank all of you in the audience who are participating in clinical trials, recognizing that we can't give you all the answers, but as we move forward, the information we derive from that trial will be critical as we move to the next one. So thank you.
I would just add that I think there are a lot of benefits to participating in a clinical trial, as you heard from some of the participants. There's a sense of empowerment that you are personally moving the ball down the field. You also get a lot of medical attention over a period of time, which can be beneficial. And then you may get early access to an efficacious drug, and I think we're seeing that in the gene therapy studies.
Okay, so there's a question. Is gene therapy a lifelong effect, or is it is it going to need to be repeated? I can answer that one. We don't know, but I think the question I'd like to pitch to the panel is sort of, how are we going to learn that answer, in terms of, what follow-up studies, animal, how are we, when and how are we going to learn that?
Yeah, I'll make a comment about that. It was deliberately why I showed the patient who was 4 years old, now exactly 4 years out post-gene therapy. Very promising long-term gene delivery for single one-time gene therapy. Does that apply to all forms of gene therapy? We can't say. Jeff pointed out a few minutes ago that we may have more problems with muscle disease than we do with something like SMA, where there's one neuron and one nucleus. We have a lot different ratio in muscle disease, but we'll find out. And but it does look promising.
And just to add to that is that we'll be following patients for years and years, and this will really be how we, there's no way else to know how long it's going to last unless we're following patients. We do have data in large animals that shows gene therapy persists for at least 10 years, and the animals are still living. So the collective data is favorable, but this is just something that we'll continue to evaluate over time, and you know, looking forward and make sure that we have the opportunity to redose if needed. So.
Yeah, I'd maybe add a few things. I mean, I certainly agree with all of that. You know, not to be overly cautious, but the data, I think, will have to come from the patients themselves, as Louise mentioned, because the animal models don't always predict things, and the vectors don't get delivered in the exact same way to the animal models as they do in patients and all that. One of the issues that I think we need to keep an eye on is that, you know, keep in mind that 40% of the human body is muscle, and there's a lot of different types of muscle. And even within a single muscle bundle, like your biceps muscle, there's a lot of different subtypes of muscle in there. And these AAV vectors don't get into every subtype of muscle with an equal efficiency. The on-off switches that are used to drive gene production, protein production, don't work equally well in all the subtypes of muscles. So one thing that I think is going to be important to monitor is, could you start to lose a gene therapy effect in a subtype of muscle but not in another type of muscle? Because with the doses that can be given safely to patients, you're not producing absolutely the perfect amount of protein in every single one of your billions of muscle cells in the body. Some are going to have higher levels than normal, some are going to have lower levels than normal. So some muscles may last a lot longer than others. And we're just going to have to see how that pans out in the patients. We do know from the animal studies, in some cases, yes, you can see gene expression out to 10 years. In other cases, if the dose is a little lower or the serotype of AB isn't quite right, you can actually lose gene expression much faster than that. So we're just going to have to wait and see and continue to advance methods to deliver the maximum possible dose.
Do we have any inkling of the difference between skeletal muscle and cardiac muscle in terms of that?
I think it's important to recognize that the trials ongoing right now with microdystrophin and then also for LGMD2E are really the first gene therapy trials for the heart at the same time. Yeah. The preclinical data supports that it should rescue the heart. The patients will be followed to see how their hearts get rescued. Yeah. In the Duchenne trials, a lot of the boys are a little too young to know whether the heart, how involved it's going to be, so it will take some time to see that. For I know at least one of the LGMD2E patients sort of getting into the age range, and then you'll find out over time, but they will look at the heart, at least, you know, in terms of MRI, look at function, see how it's responding. But at the same time, these are the first trials for neuromuscular, for muscular dystrophy. It is also the first trials for treating cardiomyopathies.
Yeah, and to follow up on that a little bit, I think there are going to be differences in persistence of gene therapy between skeletal muscle and the heart muscles. We've already seen that in the animal models. You know, one way to look at it is the interesting thing about these AAV vectors is they will persist, they will last in a cell as long as that cell is alive. But a lot of tissues in your body, the cells turn over fairly quickly. Particularly your blood cells turn over very quickly. Some of them turn over in a matter of weeks. Others, it's a matter of months. Liver cells turn over every couple of years. But the longest lived cells in the body are neurons and probably heart muscle cells. Those typically last a lifetime, and if they start giving out, you develop some serious problems in those tissues. Now, skeletal muscle is not one of those tissues that lasts a lifetime. There is constant maintenance and regeneration and turnover of skeletal muscle. It's very, very slow. It's so slow that it's difficult to measure. We don't know how long skeletal muscles are going to last. They're not all going to last the same. It's going to depend on the workload, which muscle you're talking about, how much pressure you put on it, things like that. So it's going to be interesting to see how long things last in different subclasses of skeletal muscle. But I think similar to the SMA that I think has a tremendous chance of lasting a lifetime, gene therapy in the heart may also last a lifetime. Skeletal muscle I'm a little more worried about.
So I'll just add a little bit of background. So for LGMDs, mostly the animal models that we have are just mice, or in a few cases other rodents, which have a lifespan of maybe a couple years. So if your only model is a mouse, you can see whether it lasts 2 years. You can't see whether it would last 10 years. Well, and also human and mouse immune systems work somewhat differently. Our immune system tends to be more fussy. In DMD, there are dog and other models. So really, it's hard to learn this answer without just looking at it in people. Okay, if trials are, what about non-ambulatory patients? And there's kind of two aspects to this. Can non-ambulatory patients participate in clinical trials? I mean, I know there's not as many readouts available. And if trials are done in only ambulatory patients and the treatment is approved, will non-ambulatory patients have access to it afterwards?
So the answer is, it depends on the trial, would be the easiest answer. A lot of times when you're moving towards FDA approval of a drug, they like an easy endpoint, and one of the most common endpoints is ambulation. Natural history trials should include more the entire population. And then as we move forward, I think over the last several years, there's been more forward thinking that you can do non-ambulatory patients because there are different metrics that you can look at that will allow them to participate. So I think going forward you're going to see more, but it still may be frustrating because there will be certain cutoffs in certain trials.
Just to add a few things to that quickly, I think when we're looking at our development plans, we're looking at the broad population, and I think you can get a trial or a drug approved and not necessarily study each population, but then access reimbursement, you may not get access for those patients. So in our design, we're making sure that we're being comprehensive and looking at the entire population. There's some novel ways to do that in sort of bookcasing the population and looking at older patients and younger patients. So it's certainly a good question and something that we are very we advocate for and that we're pushing for. We want the entire population to benefit.
I'll just throw in, remember you will be treating the heart, and your indication could actually be to treat the heart. And if you, at the same time, treated the skeletal muscle, that's fine. So it may actually be that you do this for the heart. For anybody who cares, our weekly steroids, we're recruiting non-ambulatory as well as ambulatory.
So I think that most investigators would like to include non-ambulatory patients. It's a large portion of the patients that we see, and we recognize that there's still a lot of function to care about and to preserve in non-ambulatory patients. I think the major hurdles still are what are the best ways to measure improvement in non-ambulatory patients, and I think we're getting there. But natural history studies, such as the one you've heard about, the GRASP study, will be very helpful in terms of our defining outcome measures for the non-ambulatory group.
I'm going to add one last thing, which is a lot of the reason that we've done what we've done relates to looking towards FDA approval, and it's very reassuring, not in muscular dystrophies necessarily, but in other disorders, so SMA and some of the ALS disorders. Even though it was done in a subset of populations, the FDA approved it for all patients with those disorders. So that's forward thinking on their part.
So I'm going to add a couple of my own questions. Does anyone care to broach the subject of how gene therapies, when they have FDA approval, can be kind of integrated into the healthcare system and convincing payers to cover them, and all of these messy but important practical aspects?
As a translational clinical scientist, I would say you hit your head against the wall and beat it down as much as you can fighting the insurance companies.
Well,
Okay.
I'll throw out something. This is purely wishful thinking, perhaps, on my part, but one of the things that I think shocks people so much about gene therapy is the current price tag that's associated with it. But it's important to keep in mind is it's a brand new therapy. The first approvals have only happened in the last year or two. Very few patients are being treated, and manufacturing is a significant issue. As the field moves forward, and hopefully we start seeing more and more successes, and keep in mind that gene therapy is not just being applied to the muscular dystrophies, it's being applied to cancer, to blood diseases, to all sorts of disorders. You're going to start seeing gene therapies coming out for a lot of different diseases. And I believe in scale of production. As the manufacturing abilities fall into place and things are scaled up, I'm hoping, but I'm not a company guy, so I can't say this for sure, I'm hoping the price tag will come down enormously in the coming years or so, which would solve a lot of these issues.
Just to elaborate further on what I said, you can accomplish the task of getting the insurance companies to yield. It may take multiple attempts, but you can. We're doing newborn screening in Ohio, and we've had very few refusals after multiple encounters with the FDAs. With the insurance companies. So it may be an obstacle. You need to face the obstacle, and then, like a lot of things in life, work a little harder and overcome it. But I don't want to leave a negative message. It can be done, and you can get it accomplished.
So Jerry, since you mentioned newborn screening, should we start doing newborn screening for LGMDs?
Well, I think that you'll meet obstacles now that you can't overcome without better preliminary data right now. So it really took the success, unequivocal success, even before it was approved that newborn screening was a viable way of potentially delivering to patients. In DMD, PPMD is making a bold attempt right now to get newborn screening, at least I would say probably not quite moving to approval through the Secretary's Advisory Committee, but making them aware of what's happening. And we did several years ago now, I can't even remember how many years ago, maybe 9 years ago? My God, I've been around for a long time. 9 years ago, we did the largest newborn screening study ever been done in the US. We did 40,000 newborns for DMD using a two-tier system where we did CK and gene analysis on the same dried blood spot. I took that to the Secretary's Advisory Committee nationally, and they said, good job, really, data really looks good. We need a little bit more efficacy in the DMD gene therapy, or in the DMD therapy world. I said we had prednisone and other things evolving that wasn't good enough. You need about 200,000 more patients and a little bit more efficacy on that side, but things are going in the right direction. So that was 9 years ago. But I think until we really have unequivocal proof, and I hope we'll have that from one of the multiple gene therapy trials that are going on now, I think we will get approval for newborn screening in Duchenne. And just to leave you with a message, I think it's not only intuitive, but it's unequivocal now in the gene therapy world. The earlier you treat, and why should it be any different, the earlier you treat, the more successful you're going to be. So I think people are beginning to, apart from intuition, beginning to accept that as a scientific fact. So you're going to see newborn screening for LGMD as soon as the Sarepta trials are successful.
I'm going to add on, I think the Secretary's Advisory Committee is going to be bypassed by genetics. And the reason I'll say that is a year and a half ago, I tried to be very controversial and ask a pediatric informaticist, when are we going to move on from 50 genes to genome sequencing at birth? And a year and a half ago, the guy didn't blink. He just said, our children's hospital is looking at 2022, 2023. And so I think it's just going to happen independent of whether or not there's approval for it, and then the data will be there, and you can act on the data.
So kind of, the other side of the coin from treating as early as possible, what if, one, has had the disease a while, what might they be able to expect from gene therapy? Would it be, plateauing of, no further progression, or might they gain some functional abilities? I realize that this is a lot of speculation on your part, but.
So one of the things that happens in all muscular dystrophies is that the muscle fibers get replaced by fat, and that fat isn't going to be amenable to gene therapy. It's really only the remaining muscle fibers that will start to express the gene that's being introduced. So we're not anticipating that a lot of function that is lost is going to be regained. There may be some small regain of function because there's some natural regeneration of muscle, but a muscle that's turned to fat is not going to turn back into skeletal muscle. So I think there are more limited gains as you become more severely affected.
Yeah, just to add to that, I mean, in animal models, we try to model this by treating older animals with more severe disease. Full disclosure, as we know, mice are not men, and it's less severe in animals, but we do try and model this in the beta-sarcoglycan knockout mouse, for example. We dosed older mice and did see the benefit in terms of expression and function that we saw earlier. So, the answer again is we don't know, and as we continue to do trials, especially in older patients, we'll be able to evaluate that, but it may be more of a challenge, as Catherine suggested.
Yeah, so I agree with both those. And, so there's some optimism and pessimism in there. Your gene therapy of a muscle cell is not going to do anything for the fat cells. Catherine is correct about that. But keep in mind, though, that the muscle, whatever muscle is left can be made stronger. So hopefully there'll be some benefit, but, maybe not as much as we'd like.
Okay, so there was also a question about cell therapies. Anyone want to broach that issue? Stem cells, either transplanted or done ex vivo gene therapy or et cetera?
Well, we get questions about that all the time, and we know, and I think Jeff said this in his presentation, that stem cells don't travel very far, not like bone marrow-derived stem cells. But in my grandiose dreams about how many years ago now? 15 years ago, maybe, we did a myoblast transfer study, and we isolated satellite cells, grew them in culture, and from donors who were immunocompatible. And I had this brilliant idea that if we delivered the satellite cells every single month, they would set up similar to satellite cells that were naturally occurring. And we did that. It was a brutal experiment, and thank you very much for participating. But we did this monthly, injected satellite cells in 50 different places in the muscle. Ooh. And did that monthly for 6 months, and we had minimal gene expression. We had a little bit, but no functional improvement. So I'm not real optimistic about using that approach.
I may just add to that, though, that you don't want to give up on potential future technologies. And if the stem cell transplants can be made to work, which is going to require a major breakthrough by some brilliant person to figure out how to solve the barriers that are out there, the advantages of stem cells is that that's the way you can grow new muscle in an older patient, is by putting in those stem cells and having them produce new muscle. So the potential is there, but right now we just don't know how to take advantage of that. But it's, that's what people in science do. They identify the problems and they go about trying to fix them and solve them. And so maybe someday they'll work, but not yet. So.
Okay, so I think that's actually all of the gene therapy clinical trial questions, but we've got a couple clinical questions. One that, I think every patient thinks about is, well, what level of physical activity, is exercise good or bad, or, how do I gauge, am I supposed to, sit there to preserve my muscles, or am I supposed to be working out, or is that going to cause more damage? You get the idea.
So I think there's accumulating data from small trials that moderate exercise is helpful. And defining moderate is really very difficult. When you have eccentric contractions, when you ask a muscle to bear a load and lengthen at the same time, that can be damaging to muscle, and your muscle may not have the same ability to regenerate. But if you avoid eccentric contractions and exercise at a moderate degree that is not painful, does not leave your muscles sore the next day, then there may be benefits. And so many of the trials that have been done have been on stationary bikes. And so, for instance, doing 20 minutes a day of stationary bike has been shown to improve quadriceps strength and walking ability. So I definitely do recommend that my patients continue to exercise.
And exercise is definitely good for your heart. So I will say there's, you want to think about the long game here, which is how do you stay healthy longer. So I agree with Catherine. I give the same advice, which is use pain as your guide. If it's hurting, don't do it. If it really hurts bad the next day, that was too much. But do try to stay active, whether that's pool activity, passive exercise with a bicycle, a little bit of active exercise with a bicycle. There are many activities you can do. Stretching, passive stretching can be very helpful. But all of that is beneficial for the heart. Moving your blood around is good for your heart.
So just to go over the definition of eccentric contractions, like if you're standing up and then you do a deep knee bend to the floor, you're flexing your quadricep muscles, but you're letting gravity overcome them. So you're stretching the muscle in the opposite way as you're contracting it. And so a concentric, which is the other thing, is that you're moving, your muscle is exerting force in the same direction that you're moving. Just a physics thing. Let's see. Is anesthesia dangerous in LGMDs as it is in DMD?
I'll just say there are many reports of trouble after anesthesia. A lot of them are in DMD, they're very dated reports. And so I think the most important thing, if you have to undergo surgery, is to, first of all, hopefully you mostly all have pulmonologists, respiratory care doctors who help look after your respiratory status. Hopefully they also have anesthesiologists that they work with. So if you're going to have any type of surgery, just make sure your team is on board. The anesthesiologist is very familiar. At Northwestern, we have a core group of anesthesiologists that work with all of our patients. If anybody's going to surgery, they not only get seen by our pulmonologists, they get seen by those same anesthesiologists. And obviously we have the cardiac team that does that where that's necessary as well. So the whole point is, as much as you can plan things in advance, make sure everybody's on board, everybody's thought through. There can be difficulty managing airway, managing all sorts of things that the anesthesiologist is in charge of. So it isn't as simple as what's the drug they're giving for anesthesia. It's really about managing the respiratory status and making sure that you haven't suppressed things so far that people aren't breathing. We do a lot of things with conscious sedation now, which is just very light anesthesia where people aren't all the way put under. And for people who don't have neuromuscular disease, they can tolerate that very well, but conscious sedation can be a big problem for somebody with neuromuscular disease. So you really need to make sure everybody's on board beforehand, that they know that you may have some breathing issues. And so as much as you can plan in advance, that's a good idea.
Yeah, I will just add as a personal note, I actually had cataract surgery last month, and then they said, "Oh, okay, here we'll put an oxygen cannula on you." Now, I knew the story about don't do oxygen without respiratory support. I don't really have significant involvement, and I was only going to be out for like 10 minutes. So I think, okay, this is probably okay, but a lot of people sort of just do things automatically, and sometimes it's going to be up to you as patients to self-advocate with the clinicians, unless you go to Northwestern. Okay, then there's a question, can an MD patient lack subcutaneous fat? So the connection between muscular dystrophy and lipodystrophy, I think in lamin C that there is a connection.
Yeah, it's probably best described for some of the lamin mutations, and in particular lamin mutations, they often fall right into exon 8. That can be associated with a lipodystrophy phenotype. So not only do these people have some muscle wasting, but then they can gain fat accumulation on parts of the body where it doesn't normally happen. And so you can see some overlapping findings there. I would say that's probably the best described case of it for lipodystrophy.
I'm going to have to go get my flight. Thank you all.
Okay, thank you. So I think actually that those are all of the questions I had. So we made it to the end with the complete panel. Let's thank everybody.